Insulin and glucagon regulate blood glucose levels. When a person eats a meal filled with carbohydrates, blood glucose levels increase. When these levels are too high, the B cells of the islets of Langerhans of the pancreas release the hormone, insulin. Human insulin hexamer. There are two unique protein chains and the protein is asymmetric. The insulin targets body cells along with the liver. Insulin binds to the tyrosine kinase receptor on liver cells to stimulate a cellular response. The phosphorylation of tyrosines on the receptor stimulate different substrate adapters like the IRS family of proteins. These IRS display the binding sites for PI3K. This activates the PKC and the AKT cascades. The activated AKT induces glycogen synthesis through the inhibition of GSK3. Insulin signaling inhibits gluconeogenesis in the liver, thus glycogen is stored and the blood glucose level decreases. When blood glucose levels are too low, the alpha cells of the islets of Langerhans of the pancreas release glucagon to increase the blood glucose level to maintain homeostasis. X-ray analysis of glucagon and its relation to receptor binding. There is one unique protein chain and the protein is asymmetrical. It is a helical conformation which is stabilized by hydrophobic interactions between molecules related by threefold symmetry. The glucagon targets the G protein coupled receptors on the plasma membrane of liver cells. Glucagon binds to the receptor which activates it and causes it to change shape. Its cytoplasmic side then binds to an inactive G protein, activating this. The activated G protein dissociates from the receptor and then binds to an adenylate cyclase, altering its shape and activity. The result is an increase in CAMP production, which leads to an increase in gluconeogenesis and glycogenolysis via pKa-dependent and independent pathways. The glycogen is broken down in the liver so that the blood glucose level is regulated again.